Assalamualaikum and good day everyone. My name is Syakirah and today my colleagues and I will be delivering a short poster presentation on pulmonary anti-infective and anti-cancer chitosan-based drug carriers. Now this poster reviews recent literature on the use of chitosan as a vehicle in pulmonary delivery of therapeutics. Before we dive into the specifics of chitosan and the various studies performed on delivering chitosan-based therapeutics through the pulmonary system, allow us first to discuss the pulmonary system itself. Now, the human respiratory system is a biological system consisting of a series of tissues and organs that are responsible for the process of respiration, with the lung being the main organ involved. Functionally, the lung is constituted of a conducting airway and a respiratory region. The conducting airway conditions and conducts atmospheric air, while the respiratory region assists in the exchange of oxygen and carbon dioxide. To deliver therapeutics to the lungs for the treatment of lung diseases, the inhalation route is favored as a non-invasive, organ-specific method of delivery. Furthermore, the cell and organelle-specific pulmonary drug delivery can be introduced by decorating the drug carrier with specific targeting ligands. The summative effect reduces the exposure of therapeutics to healthy lung cells and other systemic organs, thereby allowing reduced drug doses and alleviating unnecessary adverse effects of drugs. With the advent of nanotechnology, the nanocarrier is recently advocated in pulmonary medicine design for the following advantages. 1. The large surface area which enables interaction between the carrier with the intended lung cells. 2. The promotion of cellular uptake of particles by lung cells. 3. Decreased drug dosages due to lower enzymatic activity, thus reducing side effects. And lastly, the carrier may be equipped with targeting ligand for cell and organelle-specific drug targeting. Now let us talk about chitosan itself. Chitosan is a biodegradable and biocompatible polymer characterized by antimicrobial and anti-cancer properties. Chitosan is the most important derivative obtained by the N-deacetylation of chitin with hot alkali. This deacetylation of chitin provides chitosan with free amino functional groups that are readily protonated to induce polymer solubilization or chemically reacted and grafted to produce new chitosan derivatives with specific physiochemical and biological properties, as reviewed in this table here. Being polysaccharidic, chitosan is mucoadhesive and possesses viscous attribute that is essential in drug encapsulation, drug release, and drug absorption kinetic modulation. As a cationic polyelectrolyte, the chitosan has a high affinity for the negatively charged mucosal interface that is rich in sialic acid and O-sulfosaccharides. Additionally, chitosan is able to reduce the transepithelial electrical resistance of epithelial membranes by disrupting tight junctions between the epithelial cells, thereby promoting drug transport pericellularly and transcellularly. Thus, the potential of chitosan for use in the development of pulmonary medicine is deemed to be high. Pulmonary infection represents one of the significant burdens to healthcare systems worldwide, and this includes infection caused by bacteria, fungi, and viruses. Chitosan-based nano and microcarrier is an attractive choice in pulmonary delivery of antitubercular drugs as reviewed in Table 3. The chitosan nanocarriers in the form of solid nanoparticles and liquid nanoemulsions have been investigated for their suitability of use in pulmonary antitubercular drug delivery. Similar findings are observed in the case of cancer nanomedicine as seen in Table 4. The physiochemical attributes of the chitosan-based drug carrier are found to critically dictate the effectiveness of cell targeting, internalization, and anti-tumor action of cancer therapeutics. Small, spherical, and positively charged nanocarriers with reduced surface area to interrupt the cancer cell membrane and enhanced affinity for the negatively charged cancer cell surfaces favors cellular uptake. As the application of chitosan and derivatives is relatively recent, further research is required with in-depth analysis including in vitro aerodynamic characterization and in vivo examination of the pharmacokinetics and functionality of these particles through natural inhalation to relate to the actual therapeutic performance. We shall end our presentation on this note. We thank you for your time and attention.